Bismillahirrahmanirrahim. Hello everyone, I'm Abrar Hussain, a PhD research scholar at International Center for Chemical Biological Sciences, University of Karachi, Pakistan. Today I'm presenting my abstract entitled The Probiotics and Vegetable Oil Association in the form of literature review study. These are the contents of my presentation. I briefly introduce the probiotics field and the vegetable oil, then their association and how they affect each other. Then very brief their importance and the recommendation for future research work. So very brief about the probiotics. The concept was developed by a Russian Nobel laureate scientist Elementon Koff in 1907 and proposed that if we can change the gut microbiota with a healthy bacteria, then they can goes to increase the lifespan of the host microorganism. While the term probiotic was first coined by the Lilly and Stilwell in 1965. The current World Health Organization, they define the probiotics microorganism are a live microorganism which when administered in adequate amounts can confer the health benefits on the host. While according to International Scientific Association for probiotics and prebiotics, they define probiotics as live microorganisms that, when administered in adequate amounts, confer the health benefits on the host, which is considered as the grammatically updated version of the definition proposed by World Health Organization. Now, very brief about the probiotics field, and that these are live microorganisms. So, question arises: can we use every kind of microorganism for the probiotics potential? So, answer is no, because there's some selection criteria. For example, non-pathogenicity. There must be no antibiotic resistance in non-virulent state. They have the ability to kill pathogenic microorganism. They can also have the ability to secrete some antimicrobial substances and have the ability to tolerate the harsh condition of the GIT. So, the organism which can follow these criteria may be used for further elucidation for their probiotic potential. What are the properties or the benefits of the probiotic microorganism? They have to boost the immune system. They have anti-aging properties, which is commonly called xerobiotics. They can also use as a medicine. And currently, very extensive research is ongoing for this. They can also have the cholesterol lowering effect and also have anti-cancer potentials. Now, which type and how much microorganisms are used or proposed for the probiotic potential? So currently, the microorganisms are divided into three types: the risky strain, the safe strain, and the doubtful strain based on their probiotic potential. The safe strain are those microorganisms which have marketed and commercialized probiotics and no apparent symptom are associated with their negative aspects. While the risky strain are those microorganisms which have apparently are which have pathogenic in nature, so cannot be used for probiotic strain. But the doubtful strain, which show both positive and negative characteristics, for example, the genus Enterococcus is a brief known example, they may be used as probiotics are not based on their extensive characterization. How probiotics can work? So the exact mechanism of work is still unknown, but the proposed mechanism include they can enhance the epithelial barrier, they can promote the intestinal adhesion of the probiotics microorganism and lower the adhesion of pathogenic microorganism. They can exclude competitively the eradication of pathogenic microorganism from the GT condition and produce some antimicrobial substances like for example, hydrogen peroxide, like bacteriocin, etc. And they can modulate the immune system. And by doing all these proposed mechanisms, the probiotics microorganism can perform their mode of action. So this will brief about the probiotics. Now, what are vegetable oil and how they can associate it with probiotics? So vegetable oil are used since a very ancient time from various applications, like for example, they were used for the perfume preparation and also for used for disease treatment. And the essential oil are mostly extracted from the plants and different parts, for example, leaf, roots, bark, etc., which have some antimicrobial effects. Plant essential oil contain organic substances, for example, ester, ketones, ether, etc., which give them antimicrobial, stimulatory, and sedative effects. It is important to note that plant-based oil, which are extracted from plants, also have a proven safety in human, and they are used since various long time. They also show a high minimum inhibitory concentration against probiotics microorganism, which make them a suitable agent to make a combination with the probiotics microorganisms. In probiotics oil combination, the probiotics decrease the pathogenic proliferation, while the essential oil can kill them completely. So by using the combination of these two by multiple procedure we will discuss is, we can enhance the functionality of both oils and the probiotics microorganism. And according to literature, it is found that almost 2,600 plant essential are extracted so far, and more than 300 are used commercially for different processes in human and other industries. how this literature was performed, the study was performed. Literature's collection was performed by probiotics and vegetable oil relationship by searching different terms and Google Scholar, PubMed, Focus, etc. The review article, research paper, and the book chapter, etc. Related material were downloaded. They were studied critically and narratively, and they were analyzed while keeping the importance of the study outcome and objectives. Then both positive and negative aspects were analyzed critically, and the results were described in the presentations, and some future recommendations also included. So the literature survey showed that different probiotics oil, for example, sunflower, olive, rice, corn, brand, etc., oil are currently used for different potential for different purposes in human subject with probiotic association. So how the idea generated that we can use probiotics microorganism and oil association. So the association between the probiotics and the plant oil were developed due to the destructive ability of the plant's oil, which they have bacterial cell, and another mechanism is they have a hydrophobicity character, which are the characteristics required for a strain of a microorganism to adhere with the mucosal cell. So essential oil have both of these characteristics, and they can bind with the lipid layer and bacterial membrane. The association between probiotics and oil is varying, which depend on the applications, the methods, and the products, etc. It is very important that to note probiotic strain, viability, and survivability in the GAT condition is very much important. And it is also important that for a strain to be effective in the immune system and perform its function, 10 kilo for 7 to 10 k for 9 CFU per gram probiotics is required. But during the study, it was experimentally known that their CFU per gram is decreased during any process, for example, due to the high temperature, due to the high pressure, or due to the heat generated during the system, or due to the presence of acidic condition in the GIT, for example. So there is decrease occur in the amount of probiotics which definitely don't perform their action properly. So the scientists develop and try to identify various procedure for this, how we can overcome this problem. By that we can accurately how much we can give the probiotics to a host can perform their action properly. So they can try different approaches, but the recent approach is the use of edible oil like olive, rice, corn, etc. oil, and they can use and identify that they enhance the storage life, viability, and protection of the probiotic strain. And it is also documented that gram-positive bacteria are more susceptible to essential oil as compared to gram-negative microorganism. Now, how both of them are affect with each other. So the effects of vegetable oil and probiotics, they can improve the viability in the GIT condition. They can also enhance the tolerance to pH, salt, heat, temperature, the gastric juice, and the artificial intestinal juice, etc. They can increase the survivability. They can enhance their storage life if we are interested to use the probiotic microorganism for any industrial life or industrial purposes. They can also give them protection from the stomach acid, etc., from the bile salt, etc. And they can also increase the shelf life of microorganism when we are using food industry. So this slide just overview about the all effects which are identified so far for probiotics and oil association. The most important aspect how probiotics and oil can be related with each other is their encapsulation of probiotics microorganism with oil. So encapsulation is the inclusion of one substance in the wall of other substances. And encapsulation with different types of oil can enhance the probiotics viability. For example, it is shown that if we can encapsulate the probiotics microorganism with olive oil, they can increase 85% of survivability in the presence of gastric juice and more than 90% in the presence of intestinal juice. Similarly, it was also identified experimentally that probiotics encapsulation with canola oil can increase their viability in the presence of pH, salt, and other GAT conditions. Microencapsulation can also enhance the probiotics protection against the acid of the acidic environments of the stomach. They can also increase their viability, storage life, and help them to reach at their specific target in specific time. And most importantly, the encapsulation of probiotics microorganism with plant-based oil can help them to protect in their stressful environments and from the attack of pages also, and can also prevent them from the harmful effect of the gut acids. So what substances, a question arises, can we use every kind of oil for this purpose? No, so there must be some properties, for example, compatibility, digestibility, etc. must be keep in mind while we use any kind of oil for encapsulation of probiotics microorganism. This chart shows the effects of encapsulated material on different probiotics. So different encapsulated material will used by different researchers to check whether the viability, survivability, and every aspect associated with this association. But they can found that mostly plant-based oil are mostly important if you use them for the encapsulation of probiotic microorganism. Now, beside the encapsulation of microorganism with oils, another effects which are associated with the probiotics and the oils include that both of them can enhance each other functionality. For example, in this combination of plant oil and probiotics, and especially in case of omega-3, can enhance the probiotics viability, but they can also at the same time increase the oil stability. Similarly, they can also improve the shelf life of the products. Probiotic encapsulation with artificial oil in the presence of proteins and some phospholipid can enhance the shelf life of dairy products. They can also preserve the probiotics and they can improve their preservation. Especially it was known that at least they can improve their preservation up to 1 year in different experimental condition. They can also, the oil and the probiotics can affect their fatty acid profile, because fatty acid substances are mostly present in the dairy product substances, and they can have some anti-carcinogenic, anti-mutagenic, and atherosclerosis potential. And it is also documented that lactobacillus and bifidobacteria have the potential to convert linoleic acid to conjugated linoleic acid, which are used for different purposes in the already their stated properties. So it is documented that fortification of probiotics with sunflower or some other oils can increase the production of conjugated linoleic acid, which definitely give the anti-mutagenic, anti-carcinogenic effect to the substances. And last, probiotics and oil can affect each other profile, especially in case of resistant profile, that the oils was found to increase the functionality of probiotics like this, their auto-coagulation properties, their adherence, and their mucin degradation ability. But it is documented that already familiar with their probiotic phenomena is strain dependent. So maybe this phenomena or this capacity will change with different strains. But recently it was also reported that the induced edible olive oil can enhance the minimum inhibitory concentration of some antibiotics like tetracycline against the use of probiotics. So we can say that the oil probiotic association can change the probiotic resistance profile also. So this is what known so far, because the speed is nascent and now extensive research is not available. This is the reason why we select this topic for presentation. And the future recommendation is how the olive oil or the plant-based oil can affect the probiotic mechanism, and how they can both associated with each other. The exact mechanism must be identified. Different oil combination will be tested for different functionalities. The molecular origin of compatibility of oil with probiotics. What are the genes and what genomic analysis of these oil with probiotics association must be updated and must be identified, and the functional property of probiotics and their effects when they combine with different oils. So in conclusion, we can say that extensive research on the probiotics identified new aspect in this field. A huge number of health benefits are identified and documented very well. Among the probiotic selection criteria, it is important that the strain must be viable during GIT conditions, biotechnological processing, and industrial applications. Different techniques were developed and applied to achieve their viability and enhance its functionality, but currently plant and vegetable oil are used in probiotics encapsulation to change its antibiotic resistance profile, enhance tolerance potential, and can increase its survivability in the presence of GIT conditions and other substances. So these are some selected references from which this data was taken. Thank you very much for listening.